Welcome and thanks for coming to this edition of Carnegie Science Evening. These lectures were started 30 years ago by President Maxine Singer, and they've been going strong ever since. Carnegie Science, in case there's anyone here who doesn't know, is one of the oldest private foundations doing scientific research in the United States, going back more than 120 years. And we're very pleased and grateful to the Martin Luther King Library for allowing us to use these facilities. And tonight, we have a very special guest, Nipam Patel. He's one of the most distinguished experts in the area of evolution and development in the world. So I want to tell you a little bit about Nipam, because when he finished his PhD, the sciences of evolution and developmental biology were really very, very separate. Evolutionists depended just on, fossils and what they could see and looking at characters, but they didn't really know how those characters in animals that they were comparing came about. And developmental biologists, we didn't have genomes back then, and nobody really knew that much about what it took to make a character like during development. But Nipam was part of a group of very enterprising young Drosophila geneticists who were at work identifying the genes that affected the early Drosophila embryo. And many of these genes were starting to come into play. So while most were very, very happy just to try to work that out and try to see how an embryo would develop and what the genes did, he began to wonder, well, I wonder how, if this is still in another animal that has a slightly different morphology. And especially one of the things you could study was along the anterior posterior axis of the animal. Like, they have legs on some of their segments in the insects and other animals with segments. So he began to solve the many, many technical problems that make it actually very difficult to see if the same expression, the same gene plays the same role in nearby organisms. But he's made a great deal of progress over the years in doing this. At one point, he decided that insects were not really diverse enough, because after all, they always have 6 legs in the adults. So he decided that he wanted to add crustaceans to the mix. And fortunately, Nipam is a very good scuba diver. He used to feed the sharks at the Baltimore Aquarium, in the tank, but he had shark food, so. But he decided, and there weren't a good model systems in these crustaceans. So he went, this is while he was had his own single lab at Carnegie, the embryology department over in Baltimore. So he just went down himself to Belize and was scuba diving around in the mangrove swamps, catching all these amazing crustaceans that are down there, looking for crustaceans that vary more on their AP axis, so he'd have some good material to do new studies with. And he ended up finding many species, but he found one that's become one of the most powerful crustacean model systems that exists. So this turned out to be great. But the thing that always, I think, was close to Nipam's heart, and he apparently started, this is butterflies, and he apparently started collecting them when he was 8 years old or some very young age, and hasn't stopped since then, although his approach has changed a little bit. But now he buys collections. But he has one of the biggest and most diverse and interesting private collections that still inspires some of his research, which he's now doing even in his main lab. For example, looking at the question of how did insects' wings get onto their backs, because most of the appendages come from the lower part of the embryo. Or how do their scales, they can be clear, so how does that happen? How do they get all their colors? So he's going to tell us about the answers to some of these fascinating questions about Evo, Devo, and butterflies, and how they do the amazing things they do. Nipam?
Thanks, Al. Thanks, Alan, for that introduction. And Carnegie's been an amazingly special place to me. And as Alan said, I am interested in I'm trained as a developmental biologist, but I'm very interested in how evolution creates diversity. And yes, my lab still works on crustaceans, but I am going to tell you about my passion for butterflies as well. So I think you'll all agree that butterflies are a fairly charismatic group of organisms. So we really appreciate the sort of beauty of the colors and of the patterns that you see on the wings of butterflies. And this is actually just a little bit of my own butterfly collection. And so this really has inspired me to actually try to do research on a number of things. That and the fact that I have a huge collection, and at one point I had to justify to my wife why I have this massive butterfly collection. So I decided I should start doing something interesting scientifically, really, with them. So I have a lot of colleagues that have done beautiful work on the genetics of patterning using this group of butterflies called heliconius butterflies. What I'm going to tell you today is a slightly different story about color in butterflies. But I want to, step back and tell you a little bit about, well, why do they have these colors and patterns on them? Okay. And we know some answers to those. We know that, for example, the butterflies, the males and females use those color patterns to be able to recognize members of their own species. We know that sometimes there's sexual selection, that females will select, for example, for the males to be really brilliantly colored. But I'm going to focus on how they use these colors and patterns to protect themselves from predators. Okay, and so the main predators that adult butterflies have are animals such as birds, other insects, and spiders. And so they can actually, easily capture adult butterflies and consume them. So how do butterflies protect themselves? So there are a number of ways that they do this. And I'm actually going to just give you very briefly some examples of each of these. So a very obvious one really is camouflage, right? So you can have, for example, this moth, when it sits on the bark, that color and pattern breaks up its outline and it's hard to see against this. One of my favorite examples are these dead leaf butterflies from Southeast Asia. So on the upper side, they're very brightly colored, but of course they sit with their wings closed over their back, and their underside is this dead leaf pattern. And you can see they have the shape of a leaf and the venation pattern of a leaf and everything. Here's a picture of 6 specimens from the same species. And you can see that no 2 individuals look alike. So that way a bird, for example, will never learn which is the leaf and which is the butterfly. And so, you can see they've gone and elaborated things that look like mold spots and holes in the wing and all these kinds of things. Also behaviorally, right, these butterflies, they don't sit up in the trees. They sit where the dead leaves are, which is on the ground. But then they'll do things like orient their body and lean so that they don't cast a shadow at the same time. So they go to great lengths to avoid predation. Okay, another way that's maybe not so obvious is something called flash and dazzle. So some really brilliantly colored butterflies, like these blue morphos, which you may have seen if you've been to Central South America or gone to any of these big butterfly greenhouses where they're flying around. So the upper side of them is this brilliant metallic blue, but the underside is this very cryptically colored brown color. So what happens is that these butterflies are constantly flashing this bright blue color. And when a predator comes after them, they flash that blue color, but then they'll suddenly sit down and then they're just this, brown color. And what happens is, and I can speak from being a collector who's tried to collect them, you keep looking for this bright blue flash. And so you keep walking along thinking you're going to see them, and they're just sitting at your feet, holding their wings closed. And so then you miss them. And so a number of butterflies do that. Another thing is that some of the colors and patterns actually deflect attacks away from important parts of the body. So butterflies, in fact, you've probably seen adult butterflies flying around that are very tattered up. So as long as the leading edge of the forewing is intact, they can fly. And remember, the adult is just the one phase of the life history. So most adult butterflies are short-lived as adults, and their point is to find a mate and to lay eggs and to guarantee the next generation. In fact, some moths, for example, very large moths like Luna moths, don't even have mouth parts. They can't even feed as adult butterflies, as adults. So they're very transient in nature. So they just have to stay alive for relatively short periods of time, many species. So this is an example of a butterfly that uses a pattern to deflect attack. So you can see this is the head, but this is essentially a fake head. And so when the butterfly sits down, it holds its antenna still, or it bends them down so they look like legs, and then it twitches the back wing. And so it has this false head and false antenna. And in fact, when you look at these butterflies in the wild, many times they look like this instead. So a bird or a lizard has gotten this back end, right? And it's lost a piece of wing, but no vital parts here. And in fact, it has one more fake antenna here left to survive another attack, and they avoid being predated. This gets carried away sometimes in the tropics. They get these really long, elaborate tails. So in some places, the predators have learned to watch the direction the butterfly flies. And so in fact, these butterflies will use a 180-degree roll before they land and then walk backwards when they land. So they really try to fool their predators, okay? Another thing that they can use some of the patterns for is actually to startle predators. So the most common example are these things called owl butterflies. And so they have these giant eye spots. And the idea is what they represent, or they look like to a predator, are the eyes of a much larger animal. So you can imagine if you're a little bird and you come across these big eyes looking at you, that you're going to be startled. And so this gives the butterfly a chance to get away. You can see another example of that actually with this moth. So it's nice in camouflage, but if you startle it, in fact, it lifts up the wings and has these big eye spots. And so again, it can startle a predator for a split second so that it can get away. Okay. Another way that butterflies can protect themselves is to be unpalatable, to contain poison. So usually the way this works, and a good example, of course, are monarch butterflies. As caterpillars, they're eating milkweed, and milkweed contains a toxin that the plant uses to avoid being eaten by most insects. But the monarch butterflies are perfectly capable of eating milkweed, but they sequester the toxin in their body. So as adults, they're still toxic to birds. And so what happens is they have these very, very bright colors that make them really obvious to their predator. So birds learn not to eat them. So if a bird eats a monarch butterfly, it throws up. Okay, and throwing up is a very good single trial learning paradigm. It doesn't work well for teenagers and beer, but it works pretty well for most things. So then the bird very quickly learns, like, this is something I'm not going to eat anymore. So of course, when that happens, then something else that occurs very frequently is mimicry. So then you get perfectly palatable butterflies mimicking the unpalatable butterflies. So in the case of monarchs, there's a butterfly called the viceroy, which looks very, very much like a monarch, even though it's distantly related. And so it gains protection from predators by doing this. And mimicry, of course, is a fantastic thing. This is a row of butterflies that are called heliconius butterflies. They're all unpalatable. These are all moths that mimic them. But these moths actually themselves are also unpalatable. So both of them gain by having the same color pattern, because the birds or spiders or things don't have to learn different patterns. They just have to learn single patterns. And then this is an example where these are all monarch relatives living in Southeast Asia. And these are all swallowtail butterflies that are perfectly palatable but mimic them, and so gain protection in that way. Okay, and then in some areas, like in South America, you'll get what are called mimicry rings. So there are 6 different species here. 3 of them are palatable, 3 of them are not. And so you can imagine as a bird, you just say, well, I'm going to give up on this, and I'm not going to try any of these things. Okay, so I want to come back to camouflage, though, and talk about how you can achieve this, right? So one good way to be camouflaged, right, if you're living around vegetation, is to be green. Okay, and many butterflies do this. They have a nice green coloration and they blend into the vegetation. So here's a picture I took outside of San Francisco of this little green butterfly. And if I hadn't seen where it landed, I would have really had no hope of finding it. In fact, I actually even did see where it landed. It still took me a minute to figure out where it was sitting, okay? And so these butterflies do a very good job of blending into the background, okay? And of course, green, because the world is very green because of vegetation, is a great way to do camouflage. But how do you become a color for a butterfly? How do the wings have any of these colors on them, green or any other color? So you're probably told not to rub the wings of butterflies, right? And that's because they're covered in what are called scales. So if you zoom in on the wing, this is what you see. So the scales look very tiny to you, but they're actually quite large in the sense that they're a single cell. That cell was growing when it was a chrysalis, but the cell is no longer alive in the adult butterfly. But while it was growing, it became covered in a polymer of sugar called chitin. This is what they make their whole exoskeleton out of. It's a very tough and rugged material. But before the cell dies, in this case, it filled with pigments. So it filled with color, with pigments that make it black, brown, yellow, and so on, okay? So we know that pigments are often used for what we call the long wavelengths of the rainbow. So reds, oranges, yellows, browns, all of those colors are generally made by pigments. And we know a lot about how pigments are synthesized. So there are individual molecules that have color, and we think about them, for example, as the dyes we use to dye our clothing. So the way they work is that these molecules, and I'm giving you an example from the pigments that are in highlighters. So this yellow highlighter has this particular chemical compound in it. And this carbon ring structure is very good at absorbing blue light. So if you have white light shining down on something and you absorb blue light out, then you have red and green that are reflected, and you perceive that as a yellow color. So that's how pigments work. They're individual molecules that absorb a particular wavelength of light, and what's left that's reflected gives you a color, okay? So that's great for making a lot of colors. But it turns out that for biological things, it's very hard to make pigments that absorb red light. And so you need to absorb red light if you're going to be green and blue. But butterflies can be green and blue. Lots of things can be green and blue. How do they do it? Okay, so it turns out that they have a very different solution to this. And it's not just butterflies. It's all sorts of examples like this. So bird feathers, where you see green and blue, even the faces of monkeys, the iridophores of fish, and even human blue eyes. There's no blue pigment in any of those things. And instead, they use the phenomena of light refraction to create the color instead. So how does that work? The best way to think about it is like a soap bubble. So the soap doesn't have any color, but the soap bubble has all the colors of the rainbow. So how is that possible? And it's possible because of the way that light interacts with very thin material, okay? So here's a beam of white light, and it's actually hitting a piece of glass here. So when light is traveling through a vacuum or essentially through air, it travels at a constant speed. But when it hits other material, it slows down. And this change in speed is called the refractive index of the material. But whenever it does that, you might be able to see there's also a little reflection of the beam here. So anytime that there's a change in refractive index, some of the light actually gets reflected. And the greater the difference between the refractive indices, the more of the light will be reflected. So what happens is if you have a very thin material like soap or parts of a scale of a butterfly, the light hits it, it gets reflected off of the top surface of that layer, but it also gets reflected off of the bottom. So in that soap bubble, it's being reflected off the very surface of the bubble, but also the layer of liquid just at the bottom, it's also being reflected. But remember, light has wave-like properties. So now these waves can interact with one another. So if after the two reflections, the waves are in phase with one another, they add up and the amplitude doubles and the light looks twice as bright to you. If the waves are out of phase after the reflection, they cancel each other out and that wavelength disappears. So remember again too that white light is really composed of a rainbow of colors. And all of those colors actually have different wavelengths to them. So what that means is that when you hit a thin material like this, certain colors are going to be amplified and other colors are going to be destroyed. And that's what's going to give you an overall color. So for a long time, we've known of this equation, which is really just dependent on the thickness of the material, the refractive index of the two materials, and the angle that the light is coming. And from this, we can predict the wavelengths that undergo maximum constructive interference. And if you take out that 1/2 term, you get the wavelengths that undergo maximum destructive interference. So in this way, you can create color. But for a butterfly to do this, it has to control this thickness or the geometry of the structure quite precisely. Okay, so how thick do you need to be, or what's the size that we're talking about? So the wavelengths of light are in this area of this sort of, I'm just giving you an idea of sizes. So it's in this area between 1,000 nanometers and 100 nanometers, okay? How big is that? So often, for better or worse, we compare things to how big a human hair is, right? So human hair is about 100 microns, a typical hair in diameter. So you're talking, 2 orders of magnitude thinner than that, okay? But the butterfly has to make it in a very, very precise way to do that. And that's how butterflies make many of the colors that you see, especially green and blue. You can amplify this by repeating those layers over and over again, and that makes it even brighter. Okay. The next picture I'm going to show you is actually a moth, and in this moth, the only pigment is black. Okay, but it can make all the colors of the rainbow, right? And it does that because all of these little scales contain different thicknesses of those layers of chitin. And so then they can tune those to be whatever color they want, right? The scales on the adult are dead. They've made this when they were in the chrysalis. But in this way, they can create any of the colors. Right? So how do you make green? Okay, and in fact, butterflies and moths that are green have evolved over and over and over again. For butterflies, there's only one genera of butterfly that ever came up with a green pigment. All the other solutions are structural, but they've come up with many different interesting nanostructures that we call them to create this light refraction. So this is one of the butterflies that we study. It's called Papilio palonoris, or the emerald swallowtail. It lives in the Philippines. It actually does sit like this with its wings open. And so that green and black color blends in very nicely to the shadows and the greenery of the vegetation in the rainforest. So if you zoom in on one of their scales, this is what you see. So they have these little dimples on them that are reflecting the light. These dimples are curved, and they're a multi-layer of chitin and air. So they're repeating those layers, but they have this particular geometry. And what that allows is when white light hits the bottom of this dimple, you get constructive interference of yellow light. When it hits the edge, because of the angle of this, what happens is it retroreflects blue light out the edge, okay? And so it's yellow plus blue, which you see is green, but these little dimples are incredibly tiny, so you can't see that it's two different colors. You just see it as a green color, and so that's how they get that green. But one of the beauties of this system is that because the blue light is retroreflected, it becomes polarized. We don't see polarized light, but they do. So to themselves, they are not the color of the vegetation. So they can see each other just fine, but to their vertebrate predators, they're camouflaged. Okay. One of the neat things that we study is that this group of butterflies, in fact, can tune the spacing of those layers and the geometry of the dimple to make colors that go anywhere from a purple to a yellow color. So we're very interested in how they can do all of that. Another great example are, again, these little green hair streak butterflies. So again, they're green, but they do it in a very different way. So if you zoom in on one of their scales, they look like they have these facets of green color reflecting at you. If you break open one of these scales and look in the EM, so it's not the ridges here that are making the color, it's this honeycomb material that's down inside. But that honeycomb has a very particular shape to it. It's called a gyroid. This is a really fun story. So in 1970, there was a mathematician named Allan, and he came up with a mathematical equation that defines a triply periodic, infinitely curved surface that divides space into two non-overlapping domains. So if you were to fly through the spaces here, you would find that there were two non-overlapping spaces that were there. But this was just a set of equations he had that defined this. And then people discovered that this structure exists in the real world inside of the scale of these green butterflies. And when it's made at just the right size, it gives constructive interference of green light. And it turns out that 3 completely separate lineages of butterflies have evolved this exact same structure to solve the problem of how to be green. So it's a really fantastic solution to the problem. So those are examples, just a few examples of the kinds of structures that butterflies are able to build in order to do that. Now, of course, as soon as scientists discover these things, one of the things they tried to do is make it out of man-made material. So here's an example of trying to replicate that dimple system. So little beads were used to make these little cavities, and then they were repeatedly lined with titanium dioxide. And then they, in fact, do have similar optical properties to that Papilio palonoris, that emerald swallowtail. But this is very difficult to do, very expensive to do. It ends up using a lot of nasty compounds. And there's a lot of interest in making these kinds of nanomaterials for all sorts of things, for biomedical applications and for industrial applications. So as a developmental biologist, I became very interested in asking the question, well, how does the butterfly do this? Because the butterfly seems to do this really easily. So why don't we, instead of just using human engineering, figure out how the butterfly does this instead and see if we can use that. So there are practical applications, though I'll freely admit, I just want to know how the butterfly does this. Okay, So one of the problems, though, is that all of this goes on inside the chrysalis, right? And the chrysalis, if you've ever seen these things, are opaque. And so you can't actually see the wing growing. So the first thing we had to do is to develop ways to see inside the wing. And so this is a movie. So we've developed techniques to be able to make windows into the chrysalis. So you're looking at a butterfly that's just pupated, right? And so it's this species here. And pupal development in this species takes about 8 days. And what you're going to see is all of the wing development. So here's the wing. So it was actually made in the caterpillar, or verts. And now you see the wing veins. And as it becomes silvery, that's when the scales are actually growing. Okay, and then right towards the end of this movie, you will see all the pigment colors come in. So it then deposits the pigments and you get a completely normally formed wing. But this means then we can see all of scale development going on, all right? So then we've been able to, here's just another example, another species where we've done this with the forewing and the hind wing. And again, you can see the wing develop just fine, and we get totally normal wing development in these situations. So one of the things we're interested in is the internal skeleton inside the scale cell. So this is made of a protein called actin that forms rods, and we think is very important in forming the overall shape of the scale, which dictates some of the structural properties. So in this movie, we've actually been able to visualize now the living scales growing inside this pupal wing. And so you can see them extend out, and we can see how these structures are formed inside of the scale that generate where the ridges are and so on. Here's a slightly zoomed in version of a movie. And again, you can see the scales growing. This whole process of the scale growth and the initiation of the structural colors really only takes about 3 days. It happens really early in pupal development. So now we have some hope in being able to look and see how these structures are being made. But the other approach that we've taken is a genetic approach to try to ask this. And for this, we've used these butterflies here called buckeye butterflies. And they're very common throughout all of North America. And you might say, well, this is a terrible choice because they're all the colors I told you were made of pigments. They're browns and oranges and blacks and so on. But in fact, they're closely related to butterflies that live in other parts of the world that actually have really nice, bright structural blue and green on them. So in this sort of evolutionary tree, this North American buckeye is right here. So you can see it's stuck in the middle of many different species, closely related species that have really nice structural color. Well, it turns out when you collect these butterflies in North America, every now and then, about 5% of the population or so has a little bit of blue on it, okay? So just a little bit. And there was a woman named Edith Smith, and she lived in Florida, and she ran one of these companies that raises butterflies to actually sell to the big greenhouses that you can walk through and see butterflies flying around. Maybe some of you have been able to do that. So Edith was one of the people that raised the butterflies that would ship there. So she was raising buckeye butterflies for this, and she also noticed that they had a little bit of blue. And it occurred to Edith, what if I take the bluest ones and I mate them to each other? And so she did that for a year, which is about 12 generations, and she made them really, really blue. Okay, they're quite beautiful. And so we were buying butterflies from her sometimes for our experiments, and we bought a batch and they closed, and we like, what are these things? So we called her up and she said, oh, do you like them? I made them. And so she had done what we call a selection experiment. She had a particular phenotype that she was interested in, and she did crosses of the animals that had the greatest extent of that phenotype and built it up over generations to be extreme. Rachel Thayer was a graduate student in the lab who took on the project of trying to understand how did they become blue, what happened to them. So the first thing she showed was that if you look at the scales of these butterflies, it's not the top of the scale that has the blue, it's the bottom of the scale. And in fact, if you rub these ridges off, you see the blue really well, okay? And so what happens is that there's a bottom layer to the scale called the lamina. It's also made out of chitin. And what's happened in these butterflies, it's become just the right thickness to be a constructive reflector of blue light. But where did it come from? So Rachel found that, in fact, the brown butterflies have a lamina that's gold. So they have a very thin lamina that reflects the kind of yellowish reddish light. And so the lamina went from being about 100 nanometers thick to being about 200 nanometers thick. So that's what Edith's selection had done, is selected for that lamina to become thicker and thicker. So it became a reflector of blue light. Okay, and then Rachel went on to show that these naturally occurring species that are related to the buckeyes that have this blue and green color, that is exactly what they do. So Edith's selection experiment recapitulated what evolution had also done. So now the beauty is that now we can use this as a way to genetically map the genes that are responsible for creating the color. So the idea is that we've got the brown original buckeyes and we have these blue buckeyes that Edith had selected for. So we can cross those together because remember, they're only separated by a few generations. They're still perfectly fertile with one another. And we get what we call the F1 generation, so the children of this cross. Now, each of those butterflies contains an equal, for every chromosome, it has one copy from the brown parent and one copy from the blue parent, okay? But in the next generation, we take brothers and sisters and we mate them to one another. There's a process called recombination that allows pieces of chromosomes to exchange and for genes to sort separately. So now in the F2 generation, these are the grandchildren of these two original butterflies. Each of their chromosomes contains some fragment from the brown grandparent and some fragment from the blue grandparent. So we can sequence all of these butterflies and ask what part of the genome came from the brown parent, which one, or grandparent, which one came from the blue grandparent. And then we can also phenotype all of these. And Rachel raised up two very large families, F1, F2 families. I actually didn't know that a female butterfly could lay this many eggs, much less that anyone could raise them all to adulthood. But Rachel did that. And so what did these F2s look like? They're really quite beautiful. They're all over the place in how much of the wing has iridescence and in the hue, so the shades of blues and greens that you see on them. So if we just look at wings that have different amounts of the same color, so this one is the same kind of bluish color, but there's different extents of how much of the wing has this. We use that as the phenotype, and we ask how that correlates with the genotype of the animals. What we see, so these are all the different chromosomes here of the animal. We see that there are two places in the genome on these two chromosomes that account for almost all of that genetic effect. So there's these loci contain a couple hundred genes, but somewhere in this region is a gene that is actually controlling this. We actually know what one of these genes is already. We know that the gene here is called optics. So it's been characterized before as a gene that controls the red pattern in heliconius butterflies. And it's a transcription factor that's in the nucleus that turns on and off lots of other genes. And sure enough, in buckeyes, here's a normal buckeye. If you go and use CRISPR genome editing technology to remove the gene in the buckeye, it turns blue. So that is the gene that's controlling that, okay? But it's interesting that it's the loss of function that actually makes it blue. We don't know what this locus is, and that has a much larger effect on the phenotype. So we're narrowing in on what that gene is at the moment. But maybe what's more exciting is to look at the F2s in a different way, which is to use those, look at the wings that have the same percent of the wing covered by the iridescence, but different hues. So in this case, you're going from purple through blue to turquoise to green. And that's a difference in thickness of the lamina going from about 190 nanometers to about 250 nanometers, right? So these are genes that can precisely control the thickness of this. And we really have a priori no idea of how a cell could so accurately control the color. And remember, if it's 10 nanometers off, it's the wrong color. So how is it able to do this? So when we do that mapping, it's a little noisier, but there are clearly, and these are lines of statistical significance. So anything over that dotted line is definitely significant. Everything over the black line is very clearly having a significant effect. So you can see that there's a couple loci, and they're different between the families in some cases, that control that hue. So we're very excited to try to figure out what, in fact, those particular genes are. We don't know yet, but that's what we're narrowing down on, okay? The other thing that we can see is that there are all sorts of other things that Edith selected for. So basically, she was selecting for pretty butterflies, right? And so it was interesting to see the other things that she looked for. One of them was for the eye spots on the wings to be bigger. And so again, in our F2 generation, we get lots of nice variation in eye spot size. And we can map this very clearly to a small number of loci that control eye spot size. So that's something else we can get at. It's an interesting question for us because there's a lot of candidate genes that people have isolated that are expressed in the focus of the eye spot. But so far, when people have eliminated them in butterflies, they have very little effect on the eye spot. So this way, we let the genetics tell us which really are the genes that control this, and we can follow that. Okay, so hopefully in this part of the talk, I've shown you that there seem to be about 2 genes that control this big switch in the thickness of the scale, the basic type of scale. But then there's probably 4 or 5 loci that can control very precisely that thickness. And that's what we're interested in most, because how do you actually manage that? So that's one of the things that we've been studying, is this sort of structural color. But I'll end by telling you the other way that butterflies can avoid predators by not really a camouflage, but sort of another way of being invisible. And in this case, it's being transparent. So this is a glass-winged butterfly that I photographed in Costa Rica. And of course, I had a very hard time getting the camera to focus on the butterfly and not through the wings to what was behind the butterfly. Okay, and so it turns out that butterflies and moths have evolved wing transparency over and over and over again, right? Just like they've evolved structural color, they've found many different solutions to this. So these are just some examples of butterflies and moths that have this transparency. So the wing itself has some inherent transparency to it. So really what you have to do is get the scales out of the way, right? Is one way to do it. But if you look at these, and these are now zoomed in scanning electron micrographs of what they've done to their scales. And you can see that they've done many different things to do this. So some moths, in fact, have been able to evolve transparent scales. So they make scales, but somehow they actually are transparent. But the most common solution is to turn the scales into little bristles, into little hairs. And now the light can pass between them and go through the wing. Okay. Some unusual solutions, like this one, where the scales are smaller and then they turn them vertically. And so they're sitting straight up and light can go in between that way. Okay. So we've been studying how they turn them back into bristles. So we see that they modify the internal skeleton of the scale to give them this very different morphology. And so we're trying to understand, again, the genes that are involved in that. But one solution that's unusual but does occur sometimes is to really stop scale development. So here's a moth from Africa, and it looks basically like a chewed up leaf. And what here, in this case, these areas of the wings actually don't have scales on them. It's a bit of an unusual solution because the problem is if you make a lot of wing surface that doesn't have scales, it loses hydrophobicity and it wets really easily. So if the butterfly or moth lives in a humid place, the wings could get stuck together. So it's not a very common solution, but some animals, some butterflies and moths do it that way. One that we've studied is this polyphemus moth, which is, again, a very common North American species. Now, most of the wing has scales, but they have these 2 little eye spot areas that have no scales and are transparent. So you can see through them in those areas. But again, in this case, they actually develop no scales at all. And so you can see that here. This is in the pupil wing, and we've stained for the scales, and you see them growing in this area of the wing. But in that area that's going to be that clear eye spot, they don't have any scales forming. And so we've been able to show, in fact, that very early in pupil development, these, in the area that's going to form scales, these red cells are the precursors to the scales just starting to develop. And they're going to divide into the scale and a sister cell called a socket that holds it in place, and that's going to eventually be the scale. But in these moths, this area that's going to be transparent, in fact, never even makes the precursor to the scale. And so we're very interested. We know a little bit about how these precursors get set up. So we're interested in how the moth in this very precise location aborts scale development, so it never makes them in that area. Okay. But you may also notice that these aren't all equally transparent as well. So some of them are really nice and transparent, but others actually glare a little bit. So you might be familiar with this problem, though, if you wear glasses, right? So if you think about it, if you have a piece of glass, right, in the shade, yeah, it's nice and transparent. But if you take it out under the bright light glares off of it. So remember, it has a different refractive index. So some light is always going to reflect off of it. And so we know, right, that you can pay extra money and you can get a coating on your glasses, right? And then they don't have any glare on them. So as humans, of course, we think we're very brilliant that we figured that out. But butterflies figured that out 50 million years ago. So here, this butterfly is extremely transparent. It doesn't have any glare, and this is being photographed with reflected light. So if you look between these bristles, they have evolved an anti-glare coating. And they figured this out many times independently, and they've come up to many different solutions for this. So that's something else that we're quite excited to look at. So I want to just thank the people that did the work. And so these are all the people in the lab that have been involved in the different aspects of the butterfly transparency and structural color work. And I want to pay a special thanks really to Carnegie Science. So as Allan said, I was a staff associate at the Carnegie. And one of the things that the Carnegie really instilled in me was the idea that I should just go for whatever the most exciting questions were to me, and to just meet the challenges head on and not to be afraid to do that. And so I think, there's not many of us that try these things in butterflies, but you can see that it is in fact a great question. It's great fun for me to work on it. And I really appreciate all the mentorship I got over the years at Carnegie and subsequently the association with it. And finally, I want to thank all of you for listening to the talk, and I'm happy to take questions.
Hey. Well, now we're going to have a question and answer session, an opportunity for you to ask questions here, and people online are asking questions. And the disclaimer is here now, but the question is just, oh, here we go. Okay, Nipam, if natural selection chose a quality in a predator like a bird that would make this skill in butterflies less effective, what would that quality be?
Yeah, so I mean, one of the things, of course, that we have to be careful about is we have to think about what the predator sees. So for example, many species of birds have much better color vision than we do. So we have to also think about what they do. So one of the things is that they can be tuned to be much more accurate in their color vision. So that actually probably forces the butterfly to do certain things and to tune its colors as well. And then this isn't so much a problem for birds, but for other animals. So for example, other insects see in the UV, and butterflies also have UV colors on them. And so that's not something we are aware of when we see them, but that's probably something that the predator could use to detect the butterfly. But equally, the butterfly actually comes up with ways to create UV colors as well.
Could you comment? There must be some kind of balance, because it seems like how long have we had butterflies with these colors just from the fossil record?
I mean,
how successful over many, many millions of years has this strategy been?
It's been extremely successful. So, butterflies and moths have been around for tons of millions of years, and they've had to deal with predators the whole time. And so there's even some fascinating fossils showing that, these very ancient insects already had things like eye spots and things like that. And so they've been there. Unfortunately, butterflies don't fossilize very well. So we have a very poor fossil record of them. But this is an arms race, basically, obviously, that's been going on for a very long time.
How do butterflies' camouflage abilities compare to other insect species' camouflage abilities? Is there something special? In other words, do they have a special ability to evolve these amazing patterns that look like owls and things like that?
I think that what gives them that impressive property is that they really have evolved very elaborate mechanisms to pattern the wing, so they can create really complex patterns. So most insects don't really have wings that are anywhere near that elaborate as a butterfly. So that's given them the opportunity to create some fantastic patterns, and their scales. So one of the beauties is that they, even the scale, even if you're not making structural color, has pigment in it. But they're different. The scales, even in pigmented situations, are very elaborate with ridges and micro ribs and all these things. And any of those can be elaborated to create structural color. So they've also, in a sense, got the precursor for what you need to then suddenly switch to structural coloration as well.
You mentioned that there were places where, they sell butterflies and you can go and see lots of beautiful butterflies. And, I've passed a number of these, I've been to a number of them, but I just wondered, as an expert, could you recommend a few that are especially nice that maybe you wouldn't want to miss, if you're in that area?
Sure. I mean, the ones that I've gone to. So my family lived for a long time in Denver, and there's a great one in Denver, outside of Denver, Colorado, on the road towards Boulder, and it's a fantastic one. I would say maybe the very most spectacular one I've been to is at the McGuire Center at the University of Florida. And part of what makes it spectacular is how big it is. So it's 2 stories high, and it's actually netted. And so the butterflies behave really very, very normally, which they can do because it's in a tropical area. And so that's one of the things in some of the butterfly houses that are very small, you can't really see very normal behavior because the butterflies sort of run into the roof or run into the walls. But this place is so big that you see morphos chasing each other. You see all of these behaviors. You see heliconius butterflies going through their mating behaviors. So that's another one that I would highly recommend if you have a chance to visit the University of Florida.
Okay, well, here's a personal question, slightly reworded from this. But you've worked on quite a few different organisms, crustaceans, various insects, Drosophila. What is it about butterflies that makes them so special?
Oh, I just think I mean, part of it, of course, is the beauty. I mean, I can tell the story of how I got started, which is that when I was 8 years old, as you mentioned, I found a dead butterfly in my yard. And I actually still have it in my collection. My parents were very indulgent. My mom sewed butterfly nets for me. My dad actually, as a gift one year, had a big cabinet put in the living room that I kept all my butterflies in. And they've incredibly moved them every time that they've moved. Now, of course, my collection is where I work, and it's become quite extensive. But I think but I have to say, it's not only the sort of aesthetic appeal of them, but it's also their life history. The fact that they undergo complete metamorphosis is fantastic. People often ask, like, what's my favorite butterfly? And I don't pull out like a particularly spectacular color butterfly. I pull out these little blue butterflies, and I think they're amazing because they're totally dependent on ants to survive. So the female lays her egg next to an ant nest, and the ants raise the caterpillar. They take it up every day and protect it while it eats, and then hide it back down in the nest at night. And they do it because they get a drug from the caterpillar that they're addicted to. And the ants don't need the drug. They can do perfectly fine without the caterpillar, but the caterpillar can't survive without the ants. And so I think it's like thinking about, and that's the case with many animals. It's just they have amazing lives.
Why don't you explain how they evolved that? And the ant didn't just eat the egg the first time before.
Yeah, I really don't know. That's a great question. Like, how do you get started with something like that? But I can tell you where it's gone to strange extremes. So in some cases, though, then the caterpillar has evolved to eat the ants instead. And so again, the ant takes it in, but then the caterpillar is armor plated. It just sits there and starts eating the ant larvae. But also to keep from being attacked, the caterpillars have evolved to make a noise like ant caterpillars so that the ants won't eat it. But there's one species that then it closes inside the ant nest. And of course, now the ants are going to attack it, but it's covered in fine white scales that just break off. And so the ants attack it, and all these scales break off, and the thing runs out of the ant nest and flies away.
I guess one could use that as an example of the dangers of getting addicted.
Yeah.
Well, are there any questions from the audience? We don't have to just do these questions from the the wider Zoom audience. Yes.
About iridescence.
Yeah.
Compared to simple coloration.
Yeah. So I mentioned that in calculating the wavelengths that undergo maximum destructive interference, in that equation is the angle of light coming in. And so that's what gives what we call iridescence, this shift in color, right? So it can be a good thing. right? So butterflies can use that to create flashing patterns so that different colors are at different angles. So for example, there's a butterfly, it has this really beautiful purple flash on its wings, but it has blued from nanostructures and then red pigment, and that makes the purple. But it's very angle dependent, so it can show it only when it wants to attract a female, and it can flash this color. But you can imagine it's also a problem. So if you're trying to be green, but the color that you are shifts with angle, then the predator can find you very easily. So butterflies have actually evolved elaborate mechanisms to also defeat iridescence. So in the case of the gyroids, you might have noticed that those little facets were all oriented at different angles. So at any given angle, you're looking at the average of all the orientation. So as you move around it, you're still only looking at the average of the orientations. And other butterflies do these things where they build these elaborate ridges that means that the light can only come in straight from above and it can't come in at an angle, and they defeat that.
Other questions? Ida?
Butterflies know that they need to change their position in order to deflect or change the reflection. And in order to get away from the predators, and how do all these mechanisms evolve?
Yeah, so it's a great question. And I think it comes back to the butterfly doesn't have to know anything, right? So evolution works by natural selection. So probably, you can imagine, and in fact, I should tell a story that, like, for example, with the dead leaf butterflies, right? Like the butterfly, it doesn't need to know it looks like a dead leaf, right? It looks like a dead leaf because over millions of years of evolution, it's looked more and more leaf-like. The more it looks like a leaf, the more likely it is to survive, right? And so in fact, you can look at the evolutionary lineage that leads to dead leaf butterflies, and you can see many things that are kind of leaf-like, not as good, and things like that. So you can imagine stepwise doing that. I should point out that Nabokov, the famous author, right, he also was a lepidopterist who curated butterflies at the Harvard Museum. But he actually didn't believe in evolution. And he actually thought that dead leaf butterflies were a perfect example of why there has to be a God, because he said that evolution couldn't possibly create something so perfect. But of course, that's because we have as humans, it's very hard for us to comprehend that amount of time that it takes for natural selection to work.
Well,
start to get eyes that look like an owl.
No, but butterflies create focal patterns. So in many butterflies, right, in making wavy lines and things like that, it starts off as a little black spot. So there's a focal pattern in many species of butterflies that's in between the veins. And so in almost all cases of eye spots, it's just a continued elaboration from that. Even on the owl butterflies, so the big eye spot. But if you look at other places on the wing, there are little tiny spots. And that eye spot is just an elaboration of that one that's gotten more and more fancy over time.
You eventually get one that's perfectly shaped eye.
Yeah, no, it's fantastic, right? And it only has to look so much like an eye to work, but the more it looks exactly like an eye, the better it's going to work.
Oh, we're going to pass around the microphone just so the audience members at home can hear the questions.
Okay.
So
Oh,
this
right.
gentleman here is going to go
Okay.
next, and then raise
Good.
your hand for the next one.
thank you.
Thank you, Doctor. I was wondering if milkweed is the pesticides they're using is causing the decrease in the number of monarch butterflies. Can CRISPR-Cas9 be used to make the milkweed more sturdy so that the butterflies have someplace to feed?
Yeah, I mean, potentially. I mean, the danger there, right, is it's a weed. If you engineer it to be resistant to herbicide, then it starts to do better. People don't, and in fact, yeah, there's a lot of controversy about engineering herbicide resistance into plants, because then it means you're going to actually use more herbicide probably. So it's not necessarily a great way. I mean, I think, the monarch is a great example of what we really, really need to do is protect their habitats. And we're just destroying habitat, not just for butterflies, but for everything so quickly. If I could tell you actually another example of protecting habitat, why it's so important. So those blue butterflies that I told you that are dependent on ants, there's a particular species in England, but it was extirpated. So they'd wiped out the habitat, and probably England is a place where there are probably a few too many collectors in such a small place. And so they eliminated. Fortunately, nothing went extinct because everything still lived on continental Europe. So a number of years ago, they tried to reintroduce it to England, and they collected them in Belgium and brought them to where they knew they'd once lived. But it didn't work, and it didn't work because the ants weren't there. And the ants weren't there because the ants liked the grass to be a certain height, and the grass was too high because the rabbits had been wiped out by myxotosis. They couldn't reintroduce the rabbits because the myxotosis is still in the soil. So they introduced sheep that mowed down the grass. The ants came back, and then they could reintroduce the butterflies. But that's a perfect example of preserving the habitat. You not only preserve like a charismatic species that you're interested in, but you probably preserve tens of thousands of species that you don't even know that live in that place.
Do we have an idea of how many genes are working together to give rise to all these, wonderful patterns and wings? And, does the QTL analysis, tell you, and can you explain QTL at the level of an intelligent 8-year-old too?
Okay, yeah. So the idea is that what you're looking for, so you can have traits that are simply Mendelian, that there's like a single gene that controls that. I mean, the famous example are like some of the traits that Mendel looked at in peas. But the idea is that this is a mathematical way when many genes are contributing to be able to parse out what their contribution is to the particular, what you're looking at, like how blue they are. And so it's a way to map when there's a couple different genes interacting to do that. So the problem always is how many genes actually contribute. Probably thousands of genes are required to just even make a scale, right? So they all could have some effect, but we're asking in this particular case, what causes the variation between these two individuals? And so it is the way, for example, that we often will map traits in humans and things like that as well. So hopefully that gives you some feel for that. But probably there are many genes required to do anything, right? On the orders of hundreds or thousands to even just make the wing. But we're looking for the key ones that have a big effect on a particular particular thing we are interested in.
What do you know about when the diet or the altitude create changes in colors? Are those changes happening in which genes are activated or not into the genome? Or?
So actually, butterflies and moths are very robust against the environment changing their color, right? So if you take a chrysalis and you raise it a variety of temperatures, it generally comes out the same color, right? They need to be robust. There's some exceptions. If you cold shock them at a very particular time, they'll always turn darker. And that's a little trick that people in fact play sometimes to get sort of aberrant looking butterflies. But they do respond to the environment, but in a very indirect way. So for example, they're like the butterflies that have the dimples. There's a species in Japan. It has 2 generations a year. The spring generation is green and the summer generation is blue. And it's dependent on both the temperature and the length of the day-night cycle, but it's the caterpillar that's judging that. And then it activates hormones, which then change the way that the genes are being deployed in the chrysalis that makes different colors. So they can have that kind of indirect effect. And there are some butterflies where the different seasonal variants look so different, they were originally described as different species. Yep. Yeah, but then often they speciate when that happens. They start to look different from one another and they form separate populations. But it's not in the cocoon. It's usually the caterpillar that senses that, and then it releases hormones which act much later, days, weeks later, to change the color.
One or two more questions? Alan, would you like to do the final one?
Yeah. why do in some butterflies, the males and females look so different?
Yeah. So it's a great question. And so, many of the species, it's usually that the males are more brilliantly colored, and the idea for that is like with peacocks, that the females are, in a sense, more valuable in the population because they're laying the eggs. Males are a little bit more a dime a dozen. And so, and the females, the colors are supposed to be a way that is a proxy for the genetic health
Yep.
of the individual. But in some butterflies, there's some amazing examples, like there's a swallowtail in Africa where the males are yellow and black and very, very showy. And the females actually have a dozen different forms, each of them mimicking a different poisonous butterfly. And so this way there can be many of those, but they don't overwhelm any one model because they actually mimic many different ones. But it's all genetically set up.
Well, I'm afraid we may have to close this session on that note, but I'm sure there's an almost unlimited amount of fabulous biology out there that we didn't have any chance to talk about in the butterflies.
Well, thank you.